blast off, and Israeli hospitals preparing to launch 20 million bacteria into space in order to prepare for the medical needs of space tourism. Now, it's part of an experiment to combat the global health threat of antibiotic-resistant bacteria. And a small chip containing E. coli bacteria is going to blast off in the coming days. After 2 weeks, doctors in Ramat Gan are going to start clicking controls in a computer to mix bacteria from different chambers in that chip with the hope of overcoming antibiotic-resistant germs. And if they're successful, they'll be able to prepare for space travelers getting sick, which is apparently important since space tourism is probably going to become a very big thing. We have Professor Ohad Galmor joining us. He is the head of the Infectious Diseases Research Lab at the Shiva Medical Center here in Israel. So, first of all, tell us why it is so important to figure out a solution to antibiotic resistance as a whole.
Okay, thank you for having me today. So basically, bacterial resistance to antibiotic is a very serious, it is global, and it's really an urgent crisis that unfortunately is escalating over time. The current estimation suggests that every year, 700,000 people lost their life due to resistant infection.
Wow.
And according to a more recent WHO study, suggests that by the year of 2050, this number will even increase to 50 million people worldwide. Old bugs that we thought are already eradicated or at least under control are now coming back and becoming more and more resistant, and we often encounter infections which are no longer treatable.
Yeah, so how will this mission to space help deal with this issue?
Right, so in order to understand that, I have to say a few words about how bacteria in general actually acquire resistance.
Okay.
So that happens in two main mechanisms. The first mechanism is by a spontaneous point mutation in the genome, in a way that changes the target site of the antibiotic in the bug. This is relatively a slow process that can provide persistence to a specific drug each time it happens. The second mechanism is known as horizontal gene transfer, in which bacteria can acquire genetic elements from either the environment or from a donor bacterium.
Okay.
This foreign DNA element is highly versatile, it's modular, and it can carry multiple antibiotic-resistant genes that can confer bacteria to become resistant.
Okay. All right, so again, space. Talk to us about space and how this plays into the issue. And obviously, I mean, it's just interesting that, you know, tourism in space is going to become this big thing, so that's where they're focused.
Okay, so there are basically two main motivations for this study. The first motivation is really the expectation that in the not that far future, more and more people will travel to space. Now there are only few people who are traveling. For example, there is the International Space Station, and people are traveling there. But in the not that far future, more and more people will explore the space tourism and will go there. And when we're sending people to space, we're sending actually more bacteria than we're sending human cells. And we still don't have a very good information about how bacteria behave in space and what are the chances on what are the risks that these bacteria will acquire resistance. So this is the first motivation of this experiment. The other motivation is that we already have preliminary data suggesting that under microgravity conditions, the ability of bacteria to acquire resistance via horizontal gene transfer process, which I mentioned before, is much, much slower. And we aim to understand how and why microgravity affect this process. We hope that when we gain this information and understand what is the effect of microgravity on the ability of bacteria to acquire genes from external source, we will be able to develop different approaches and different tools to prevent bacteria from acquiring these antibiotic resistance.
All right, well, listen, you broke this down in a very easy to understand way. Thank you so much for joining us. Super cool. Let us know if you figure out a vaccine for coronavirus along the way, all right? Okay.